Hello, everyone. Welcome again to another session of the Bioconductor Africa Seminar Series. Today we are going to have a session on multi-omics data integration with Bioconductor, and we are very lucky to be joined by Dr. Mallick and Nalin Arora from Cornell University. So before we start, I'll just read a few housekeeping rules to tell us how we'd like to interact during this session. And in case you've not seen it, that's on line 15 of the Pad. We do have a code of conduct, and we expect you to treat people with respect and not intimidate them or bully them or harass them. And in case you experience any of these things, please write a message to me here on Zoom, or you can also email me. So this call will be recorded and it will be uploaded on the Bioconductor YouTube channel. So if you'd not like to be, identified using your name or your image, please anonymize yourself. You can use a pseudonym. You can use an emoji to make sure that you're not recognized through the recording. If you do not anonymize yourself and later you feel like you need to, you can always write to me and we can remove any identifiable characteristics from yourself on the notes or on the recording. So notes will be taken on the pad that's been shared on the chat. And again, if you've not taken part in the roll call, please do so. It helps us with record keeping and just knowing who's on the call. In case there are any AI note takers, they will be removed for privacy and safety reasons. Also, this call will be uploaded on YouTube, and the notes will be archived on the Bioconductor Africa Seminar Series website, so you can be able to find them later. Yeah, and I will introduce our speakers today. We have two. This is the first session where we're having two speakers, and I'm very excited to see how that goes. So in case you'd like to read their bios, that's on line 51 of the Pad. We have Dr. Emil Mallick, who is a tenure-track principal investigator at Cornell University. He's the creator of the widely used open-source statistical software and multimodal AI tools, most notably Maslin, Bioconductor package that has become a community standard for microbiome, multi-omics association analysis. And you can read his full bio on line 51. We also have Nalin Arora, who is a master's student in biostatistics and data science at Cornell University, and he works with Dr. Mallick in the Department of Population Health Sciences. His bio is also available on line 55 of the Pad. So I will hand over to Dr. Mallick and Nalin, but I would just like to confirm if they will be taking questions after the presentation or during, so they can tell us that. Welcome.
Yeah, I think during is fine. Feel free to ask questions anytime. So I can share the screen whenever you are ready. All right. Okay, just one more confirmation again, if you can see my slide.
Yes, we can see them.
Okay, great. All right. So first of all, thanks for the invitation. And this is our first time with the Bioconductor Africa community. So we are really excited to talk about this topic, multi-omics data integration with Bioconductor. So before I go further in terms of the details, I just want to mention briefly that, as you possibly know, multi-omics data integration is a pretty broad topic. And what we are trying to do here is we are going to focus on one software. It's called Integrated Learner. But before we jump into Integrated Learner, we also need to know what are the basics of multi-omics data integration, especially with respect to some of the terminologies that have confused people, especially those who are new to the field. So the part that I'm going to cover for the first half or so is some of the basic concepts related to multi-omics data integration. And I also must mention that I typically use the term multimodal because even though multi-omics is much more popular, there are some folks, I'm sure there might be some in the audience as well, that they may not have a omics data, but they may have multimodal data. So multimodal is much more general. So we are going to use that term multimodal and multi-omics interchangeably throughout this seminar. Okay, let's get started. And very brief introduction. I think we are a pretty new lab at Weill Cornell Medicine at Cornell University. So those of you are not familiar with the Cornell campuses, Cornell have two campuses. One is in Ithaca, which is 4 hours away from New York City. And we are in New York City, which is in the medical school, Weill Cornell Medicine. And one of the lab's research agenda is multi-omics data integration, and we are actively developing software tools, including integrated learner, which you are going to learn today. And Nalin is a developer of integrated learner. He recently submitted integrated learner to Bioconductor, which is not yet a Bioconductor package, but it's currently under review, should be in Bioconductor soon. But he extensively worked on integrated learner and some other related tools. I wanted to just briefly pause here so that Nalin can quickly introduce himself.
Hi everyone. So I'm Nalin. I have been working with Dr. Mallick for a while now. I think my starting project was integrated learner, and post that I've just been working on building softwares that include that basically utilize integration of multiomics in the different areas. And I am currently pursuing my master's in biostatistics and data science with a possibility of doing a PhD down the line. Thank you.
All right. Thanks, Nalin. Okay, so let's talk about quick logistics. And this is something we haven't shared because I think it was pretty much last minute, and I was also on vacation until August 10. But I think one thing, if you can, and that will be really helpful, is during the lecture session, or at least the portion where I will be covering, you can, if you, again, if you have everything available in front of you, you can go to this GitHub repository that we have created, and you can possibly do the cloning. And there is an install script within that repository. You can simply run that script once. I mean, if you are lucky and if there are no issues with any of the packages, which we cannot guarantee, of course, because there are so many dependencies. But if you are lucky, there is a chance that it will just finish installing all the packages at once. So I'll request you to do that just in the background, because Nalin will be doing the tutorial in a few minutes from now, while I will be going over some of the concepts. And another thing that we are going to use this two data set that you see here in the screen, HMP-IBD_TSE, which is a tree summarized experiment object. And then there is a summarized experiment object, which is another data sources. And we will see how we can use this data set for, firstly, to pre-process, how to make it bioconductor friendly, and then how to use integrated learner on this data set. And possibly more examples from our software tutorial if we have enough time. But given that we have 90 minutes, I would possibly start with this, and then we'll see how things go. Okay. Sorry, there are some chats. Yeah, so if maybe for everyone here, what I'm going to do is I'll post the, is it possible to paste the link? Yes, I'll just paste the link here, just so that everyone can go and clone that repository. Okay. And after you have done, you are done with the installation and make sure that all the data files are in place. That means the cloning was successful. Just be prepared with a RStudio session, because Nalin will be doing the tutorial on how to use these data sets for a multi-omics integration case study. And feel free to ask me for any questions. I will be checking the chat possibly, and do let me know if anything is unclear. So to set the stage, I will start with this really interesting figure from a recent paper, which showed how we are already in the age of multimodal AI or multimodal data integration, and possibly we can also use the word AI-driven multimodal integration, simply because of LLMs nowadays can also generate different sources of data just using NLP and other techniques. And all of these data together is obviously multi-omics data. I'm sure that those of you signed up for this seminar are already familiar with multimodal and multi-omics data. But the point is that in this setup, there are the goal that was already there in the single omics or single view setup remains the same, especially for the biology. If the goal was previously better identification of maybe a patient subgroup, the goal remains the same. Can we do a better job with multi-omics data? If the goal is to predict a disease outcome or some sort of predictive modeling, again, the goal remains the same, just that we want to do better job as compared to a single omics or single view. And those are reflected here in this particular figures, that we ultimately want to do better biomarker discovery using multiple types of data, because biology is complex. And nowadays, with the AI-driven or LLM-derived features, we can possibly do a better job. So that's the hope. And I will simply simplify this using this cartoon of elephant. So if you see this elephant, if you think from a single omics standpoint, we are mostly looking at a different part of the elephant and possibly concluding relatedly based on our observation on based on that single omics. But multi-omics, the hope is that we can possibly take a broader lens or a lens that will give us a comprehensive view of the elephant. In this case, obviously, that is any biological outcome of interest or biological phenomena of interest. On the left, you will see that the goal of, to do predictive modeling so that we can say, the one example that I want to use is responder versus non-responder, which is pretty typical in a treatment setting, like who is going to be a responder versus non-responder. You can also think of any case versus control, healthy versus disease prediction, which can help you in subgroup identification. Here also, as I mentioned previously, the goal remains the same, that we want to use the multi-omics data in order to do a better job at yes, the slides will be shared just to answer that question. So we want to do a better job using this multi-omics data. And here on the right bottom, I have actually summarized it more from a machine learning and statistical language standpoint. So if you are already familiar with traditional machine learning, where you have possibly a high dimensional X, which is basically any omics data, we now have a little bit complex scenario that we have multiple such X's. So I usually use this term from X to X1, X to Xk. K can be up to 10 or 20. I think I have not seen anything more than 10 as of yet. That means number of modalities being more than 10 is uncommon, but obviously the way we are generating data using AI and other techniques, very well, it could be more than 20 in recent future. So that's how I want to summarize the typical difference from a single omics or single view modeling from to multimodal modeling. We now have multiple X's as opposed to a single X. Remember that the single X was already complex enough from a omics standpoint, that it was already high dimensional. We already used to have machine learning model that was capable of modeling that high dimensional data. We have been, again, I'm thinking or mostly stating the historical perspective, even from a single omics traditional machine learning standpoint, overfitting, high dimensionality, and so other challenges we wanted to resolve using sophisticated machine learning techniques, we now have to apply the similar concepts in the setting of multiple X's. So that's the gist of what we are going to talk about today. So this is an example multi-omics study. Example being, this is a real multi-omics study that concluded in 2019 and was published in a Nature paper. The reason I'm highlighting this, there are other multi-omics studies as well, especially I saw that the next seminar series in single cell. So in single cell, there are many single cell multi-omics studies, as some of you are possibly familiar. But I'm highlighting this one because I have been involved in this. And also this is one of the largest microbiome multi-omics data, which has been one of the research area for many years in the lab. And in this particular case, if you see the total number of subjects, those are really close to 100, but the number of features that have been collected and number of data modalities that have been collected, and also number of samples that have been collected, that means multiple repeated measures for many of these modalities, those are on the higher side. So more features, more data points, but the typical number of subjects are on the lower side. So this is one of those early multi-omics study. Here, IBD is the main outcome of interest, and this is also the data set you are going to use in the hands-on session that will be led by Nalin. In this particular study, the goal was to see IBD, inflammatory bowel disease, how that is impacted by multiple modalities, in particular microbiome and metabolites. There are also other host transcriptomics and other clinical data, for example. That's the reason, as I said, multimodal is a better terminology than multi-omics in general, because it covers many of the non-omics modalities, for example, clinical variables, as part of the umbrella term. Before even thinking about multimodal integration, so one of the first thing that you can do is, if you have two modalities, in this particular example, even though you can see there are so many modalities, microbiome and metabolite are the two main. So if we focus on those two, and one of the easiest thing, even before thinking about integration, you can do is just cross-correlate this microbiome and metabolites. This is what has been done in this very nicely generated figure and heat map, where on the x-axis, you can see the metabolites, and on the y-axis, this is basically the species or microbial species. And some of these correlations simply mean how they are associated within the individual IBD groups. Even such a simple correlation can also lead to some actionable discovery. So one of the example in this particular situation is these two associations that we are highlighting here were also experimentally validated based on a population level, data-driven correlation that was highlighted in this heat map. So even a simple correlation analysis of two modalities, just simple feature by feature, it's also called all against all association, can also reveal a lot of things you want to know about multi-omics. So that's one way to approach multi-omics data. And I must mention that there are actually a lot of details that I have not mentioned. For example, this particular figure was generated based on residuals. That means for each of this feature, some of the confounder that are going to impact both metabolites and species were subtracted. That includes antibiotics and some other treatments. Maybe age is one of the confounder as well. So in that sense, this association is much more deconfounded, because both the species and metabolites have been residualized to take out the effect of any confounder, so that this association is much more real, and that was reflected when the experimental validation also worked in this particular case. Again, that's not the only way you can associate two modalities. Another way that people do is also very common, is before even thinking about which feature is associated with which feature. So in the previous example, this is more about feature to feature. So each point is a correlation between one feature in modality 1 and another feature in modality 2. We can also ask the question, is there any association between the two modalities at all? In order to do that, there is a test called Mantle test, which is becoming very popular. This is important because if there is no association at all in the data that you have collected, there is a chance that no further analysis is going to be useful. So Mantle test usually works as a first-hand check to see if there is any association at the data level, not at the feature level, at the overall level, between the two modalities that you are interested in. The way it works is it calculates the similarity matrix for both dataset X and Y. So remember that if here the assumption is that you have the same number of subjects or matching subjects or samples across two datasets. So that means whenever you do this similarity calculation, which is patient by patient or subject by subject, this becomes comparable. So if you don't have same number of features in dataset X and dataset Y, with the similarity matrices, you now have same number of subjects because it's an n by n similarity matrix if you have n number of samples. Then you can simply vectorize that similarity and calculate correlation and get the significance using permutation tests. So that's the idea of the Mantle test. And the previous IHMP study that I just mentioned, it's also called IHMP and HMP2 again interchangeably. It's Human Microbiome Project Integrated and also Part 2. So both of them are valid. And here, what they showed in that Nature paper that there is tight coupling between some of the modalities, but that's not necessarily true for some of the other modalities as well. So that gives you, after you have collected your multi-omics data, this is what will tell you which modalities are tightly coupled and connected and which modalities are not really coupled together. So that may give you some sort of idea before you do your multi-omics integration. I see a question in the chat. Okay, that's a comment. Great. Okay, so one might say that the Mantle test is not going to give you what you need for more actionable inference, for example, feature by feature. So we already saw that you can simply do, as we saw in the previous slide, that all versus all association. That can be a little bit tricky when your number of features grows exponentially. And that can happen that if you have more than two modalities, that is obviously going to make things worse because you have so many feature by feature combination. So in order to tackle that, one of the way people are doing it nowadays, and some of you might be familiar with the MOFA, which is the multi-omics factor analysis. So the reason I'm introducing all these methods is because these are the basics, or even before thinking about integration, you can do this, how these modalities are associated with each other. That typically gives you some hint towards what you want to do later on. So one of the way you can do feature by feature association is just projecting the data into a lower dimensional space, something like PCA. Here, PCA is a general term. It can be your favorite ordination algorithm. It could be based on PICOA or other dimensionality reduction technique factors as well. So the goal is you reduce your high dimensional data to a set of factors and then use the factors for doing all versus all correlation. Obviously, this gives you some advantage with respect to computation because you are really comparing less number of pairwise associations, but then you have to trace back how the individual features are associated using the loadings, and there are multiple approaches based on loadings mostly, and how do you connect back to the original feature space. That's a problem, but typically this works very well because with more than two modalities and if a handful of modalities, this approach gives you the feasibility of looking at how things are associated with each other, especially on a feature by feature basis. And one of the earliest methods is actually came out in 2014. Even I think this might be one of the methods that is ahead of time. Multi-omics data was not that popular by that time, but they came up with this similarity network fusion, which uses the idea of constructing the similarity networks is again very similar to what we just mentioned in the mental test, that you want to use your omics data, individual omics data, not really doing any integration as of yet, but convert that into patient similarity matrices by just simply considering patient by patient similarity. You can use any measure for this similarity. It could be correlation, it could be any other similarity metric. So once you get this patient similarity network, the idea is, can we fuse these networks based on the individual modalities to come up with a consensus network? And that's the idea behind this similarity network fusion. What they showed in this paper that was published in Nature Methods in 2014, that when you have individual modalities, you have pretty noisy representation of the clustering that you are hoping to see in your patients and subjects. But when you do this fusion, you see that heat map here, pretty noisy, A, B, C, but D, it's a pretty clean heat map. So you can now clearly see the subgroups, which was not evident from the individual omics. I will warn you that this is a very highly optimistic figure. In the real data, you may not see that exactly, but for this particular data type, in this particular paper, that happened to be the case. And this is also one of the way you can integrate multiple omics modalities. And here is a case study based on a real data that is not from this paper. So this is a study on autism spectrum disorder on children, where a particular treatment called microbiota transfer therapy, or MTT, was provided. And then there was a comparison group as well. And it was more like a pre and post setting, where you obviously collect the microbiome and other modalities at day 0, and then also after treatment, in this case, after 10 weeks, as well as after 2 years. One of the goals in this study was to see that the comparison was with respect to the normal child, which are called neurotypicals. So the idea was, if the treatment has worked, the ASD children will be very similar in their multi-omics profile as compared to the neurotypicals, which is also called the TD group. What this SNF analysis revealed is that if you do this SNF similarity network fusion, all the multi-omics data that was collected, so in this case, microbiome, stool metabolomics, plasma metabolomics, there are other type of data as well, that shows that there is a definite 2 clusters revealed by SNF. So that means that at baseline, as expected, the treatment is not really, there is no treatment yet. So that means the ASD and the TD children, there are 2 clusters as supported by the SNF. But when it was done at the post-baseline, you will see that the algorithm did not find 2 clusters. It was mostly all of them in the single category except 1 patient. This was a way to show that these ASD children are now becoming more close to the TD children after the treatment. This was done using this unsupervised technique. So remember that we are not using, so far, all the examples that we have talked about, any outcome information in our association, be it an SNF, be it a mental test. We are only using the individual omics and trying to combine them together. And that is actually going to lead me to the next section. So all these things that we have talked about so far, SNF, mental test, all versus all as correlation or association, they are all considered, including MOFA as well, are unsupervised. So that means we are not really looking at a particular outcome of interest, and we are not doing any supervised analysis. We are simply doing a unsupervised analysis, trying to integrate the data, which is a whole body of literature. And there are several factor-based and multi-omics factor-based models that are always coming up, which are trying to improve how to do unsupervised clustering, similar to this SNF framework, based on multi-omics data. That is not what we are going to mostly focus on today. What we are going to focus on is, if we have an outcome of interest, how do we do multi-omics integration with that outcome in our mind? And that typically works well because unsupervised clustering is obviously useful, but we are mostly interested from the standpoint of biomarker discovery. So what are the features from the multi-omics are associated with a particular outcome of interest? This is also reasonable because in order to do biomarker discovery, you need to know which features are predictive of the outcome, and so on and so forth, based on your integrated multi-omics profiles. One of the things that has been a little bit all over the place in the multimodal integration or multi-omics integration is that the vocabulary of how to integrate the data was not very clear until very recently. So we recently published this book chapter trying to address this gap. What we really mean by multimodal integration. So far, we have talked about unsupervised multimodal integration. Of course, that is a very solid terminology. That means there is no ambiguity there. But still, we didn't really formalize the definition of how we are doing the integration. So we'll see that there are actually multiple ways you can integrate multimodal data. And even you can define multimodal data in different ways. The particular example that we have just talked about, that means same set of patients, same set of subjects. For them, we have collected multiple omics modalities. That is a typical scenario that you can think of, and possibly that comes to your mind naturally, that might be the only multi-omics integration scenario. So vertical integration, which is the most popular form of integration we have so far, all the examples that we have talked about, is the same set of patients giving or having multi-omics features. That means multiple modalities. And that becomes our shared anchor. So this is a terminology, what connects the data. Here, what connects the data is the subjects. The patients are the common factors between these modalities. That's why we are integrating all these omics modalities on the shared subjects or the same set of subjects. So that's the whole goal of vertical integration, regardless of whether you are doing supervised or unsupervised. There are two other types of integration, which typically we don't talk about too much, but is also becoming more and more common and popular as well, because many of the fields are moving so fast that not only we have same set of subjects, we may have different set of subjects, but the same number of same features across multiple modalities measured between them. So this will be an example where you have multiple studies, maybe on a particular disease of interest. So one example that I typically use is meta-analysis, where you are same outcome has been tested using same set of modalities, but in very different heterogeneous study populations. So one study could be in the US, another could be in the Europe, and another could be in Africa and Asia, and so on and so forth. And there might be interest in finding what are the most consistent features that are possibly aggregating all the studies we can discover. So that type of integration is called horizontal integration. So here, the shared anchor is the features. The reason you can even think about combining these multiple studies or groups, because you have confidence that the same shared features have been measured across these different studies. So that is also a multimodal integration problem. In this case, the multiple modalities could be multi-omics or single omics, but they are definitely coming from multiple studies and different set of patients. And your goal is to combine different set of patients, similar to a meta-analysis setup, to find the most consistent features associated with an outcome of interest. And there is another category, it's called diagonal integration, and this is even less developed or less popular. But there is I have a slide where I have a review paper that you can check that in single cell, this has been a pretty popular topic, which basically means that you may not have a fully resolved shared anchor. This can happen, for example, you may have a single cell proteomics. You may have a single cell transcriptomics, and maybe this could be something like a bulk RNA-seq or microbiome data at the subject level. So if you think about integration in this particular setup, you will see that even though there are some subject level shared information, your data is at the cell level for omics 1 and 2. So that means cannot really, like, you are not in a situation that you are ready to integrate the data, as opposed to the vertical integration when you had the subjects as your shared anchor. Here, because there is a hierarchical within subjects, there are cells, and the cells may not be corresponding to each other. And in another case, there is no cell information, for example. That means that we are in a diagonal integration setup. And this is just one setup of diagonal integration. So diagonal integration means anytime you don't have a perfect match with respect to either the features or the samples that you are trying to integrate. In most cases, it's the feature level, the subject level mismatch or sample level mismatch. That is the more popular and common one. But it can also happen at the level of not having features that are agreeing between multiple studies, or same study but use different feature sets. So how do you align those features? That is also a diagonal integration problem. In the horizontal integration, again, remember that we did not have any problem with the features. We assume that the features are shared. So if it is gene 1 remains gene 1 in study 2, and it remains gene 1 in study 3. What if I have specific example, but I don't want to go into typical the details here, because in the microbiome field, there are different types of data that you can collect. For example, 16S and shotgun metagenomic. Even though the studies are different, you may have features that are not exactly aligned. So you may have to come up with some solution to match the feature names or features before you do your integration. So that's from the side of features, but mostly it happens on the side of samples. And in the IHMP example that I mentioned a few slides ago, remember that in this particular data, this is also multi-omics, and you'll see that there are some differences in the number of samples. This is simply because you have longitudinal profiles for some of this. That means it has been collected longitudinally and it was feasible or possible based on the protocol, which was the case with most of the microbiome and metabolite profiles, but it was not the case for other profiles. In some cases, it was only cross-sectional, so it was only collected at one time point. That would be also an example of a diagonal integration. So if you remember again that the mismatch that we were talking about, this could be your longitudinal omics, this could be your cross-sectional, and this could be your another longitudinal where the times are not exactly matching. It could be week 1, week 10, and so on, and it could be week 2 and week 10. Anytime we have some sort of mismatch with respect to the samples or features, that's a diagonal integration problem. It's an open problem. There are methods that are being developed. And obviously, we are going to focus on today on the vertical integration, and I will have some materials to cover the horizontal and diagonal if we have time. Okay. Let me check quickly if there is any question. Okay. All right. So let's talk about vertical integration. And even though vertical integration as a concept, you may have already figured out, okay, this is the vertical integration. That means I have a perfect setup of same set of patients or subjects, and I have multiple modalities. Even within that setup, we have multiple ways you can do the integration. So this is summarized again in this figure. So early fusion is the most popular and most, I think, simplest, and possibly everyone is familiar with it. It's basically concatenating all the views that you have into a one single view or matrix or data frame, what you want to call it, and then do your regular machine learning or analysis. That would be called early fusion. The reason it's early because you are fusing the data at the very beginning pretty early in the algorithm. The second is the late fusion integrated learner that we are going to talk about is a late fusion framework, although it does incorporate early fusion as well. Late fusion builds on the idea that if you want to build individual models from the individual views and you don't want to mix them together simply because very different signal to noise ratio exists between in these views, and they might be if you combine them together, there is a chance that you are blocking some signals to pop out simply because one modality completely dominating the other. So in that situations, and also that's the philosophy behind this late fusion, that you build these individual models first to predict your outcome, and in a second stage you combine that predictions to get your final prediction. Both early and late fusion have their advantages and disadvantages. Intermediate fusion is again another emerging topic that is coming up in recent years, which basically believes that because we are in a multi-omics integration setup, we should be utilizing the shared common biology that exists in the multi-omics data. So the idea is we want to come up with some sort of latent feature space or augmented space. This is done using factor analysis or some other techniques, where before you do any machine learning, you simply want to extract some common factors and any way you want to incorporate the dependency between the two views, and then you do your machine learning on the augmented space. And intermediate fusion is typically, generally, based on multiple papers, are considered the gold standard now, although not too many methods are still there, but people are going towards this simply because early fusion and late fusion are too simplistic. Intermediate fusion gives you that balance that you need. That means you want to capture the dependency using something like a factor and some other way that will reduce the noise and only capture the complementary biology. And then you do the model building, which is neither early nor late. So that's why it's called intermediate fusion. As you may have possibly guessed, that early fusion remains the most popular, and it's pretty widespread in the literature, including many of the so-called multimodal AI methods currently being developed. It just fuses all the data together, and then it tries to do the standard machine learning on the fused feature. So early fusion remains pretty popular, and it's not always the case that early fusion is the winner. We'll see that which is the best fusion paradigm is not 100% clear based on the current literature. I saw a question, maybe I'll try to answer that. So the question is, how do you perform loadings in PCA? So we don't perform loadings in PCA. Loadings is a term where when you do the PCA, you have the coefficient. Like if you remember PCA, that you are trying to come up with a linear combination of features in the form of PCs or principal components. Those coefficients, how they are weighted in the PCs, those are called loadings. So using those loadings, you can trace back. So if PC1 and PC2 is associated, within PC1, you have the loading for feature 1, feature 2, and so on. Using the loading strength, you can say which feature is contributing to the particular PC. So loadings is part of the PCs, which is the weight of the individual features on the PCs. Okay, so I'll briefly introduce integrated learner here, and I see that the time is almost 45 minutes. So I'll let Nalin start his tutorial so that you can get a sense of how to use this integrated learner package, which is going to be a bioconductor package soon, currently under revision. So the idea of integrated learner is that it obviously uses late fusion as its main default algorithm. So here you see that if you have multiple omics, these are some of the things you can ignore for the moment. You have to do your pre-processing. So left-hand part of it is more about how to do the pre-processing. And then once you have the pre-processing done, integrated learner uses the concept of base learner. Base learner simply means omics-specific learner. you can choose many of the options that is available. So this package is built on another R package, very popular, called Super Learner, which includes more than 50 algorithms. So you can use any of the base learner as your choice, like random forest and XGBoost, so on and so forth, for each of the individual omics modalities. And then you also need to choose the meta learner, because if remember that in late fusion, you have to combine the individual level prediction in a second stage machine learning model. And we provide that information, user-defined options to do choosing base learner, choosing meta learner. And you can also say, I want to do early fusion, which is simply, there is an option to say that I want to do both late fusion and early fusion and see how the performance is. But the most important part of integrated learner is it actually is a generic framework. It can now model survival, multi-class, continuous, and binary outcome. And you will see that in the tutorial, how to do that using bioconductor friendly containers like multi-assay experiment. One of the things that is really important in the context of machine learning is that, because remember that in this integrated learner late fusion framework, you have to do your predictive modeling based on each individual omics. So that means some sort of cross validation is going on in omics 1, omics 2, and omics n. If you don't do the cross validation carefully, there is a chance of data leakage. And this is not something that typically talked about in early fusion, because that's not a problem in early fusion. But in late fusion, it's a problem that you have to make sure that the way you do the cross validation, that remains exactly the same across the omics. The same set of subjects or same set of observations are typically held out for each fold, so that when you do this prediction and combine it across multiple omics, you don't have a data leakage problem. So only when you do this in this manner, which integrated learner does, you can do the final prediction using a very simple weighted combination. Another thing that I want to mention, that even though in principle you can use any machine learning algorithm for your meta learning step, that means if you have 10 modalities or 5 modalities and you have built 5 random forest models, you can simply combine the 5 predictions using another random forest. That has been the case with some of the existing methods, that they tried to be fancy in the meta learning step, but integrated learner actually tells you to be not fancy, because at the meta learning step, the problem is very simple. You have only k number of features, so not too many. So that's why you recommend this least squares approach, which is another way to say weighted combination of features using non-negative least squares. The reason it's non-negative least squares, because we want to provide these weights such that it's between 0 and 1, and also it's sum to 1, so that you can interpret it as a percentage. So 10% of the prediction contributed by microbiome, 20% by single cell, so on and so forth. So it's highly interpretable and highly flexible as well. Okay, I'll quickly finish this case study. So it's a published paper already. If there is not a link, I'll possibly add it here at some point. It's a published paper. You can see multiple case studies. The one case study that I want to highlight is this: a continuous gestational age prediction from this 17 subjects, pregnant women. There are 3 time points each at 3 trimester. And the goal was to, again, see, can we do gestational age prediction well based on multiple omics modalities? And this is based on another 2019 bioinformatics paper. And what we showed there is that if we do the integrated learner way of doing the integration, where you have a base learner, so this particular x-axis is underscore base learner underscore meta learner. So NNLS, which is the non-negative least squares, which is the default. If you use that individual base learner and the NNLS combination, that usually performs the best as compared to doing some fancier version. So for example, this particular paper, 2019, it uses ElasticNet as your base learner and ElasticNet also as your meta learner, which is not needed, and that heavily compromises the performance. And these are some, this is Franzosa et al., which is basically an early fusion. So this basically shows that 2 things. One is sum is greater than parts, which is part of the reason that we are doing multi-omics. And also integrated learner is better than state-of-the-art methods. And when you run this, this is what you will see in your console, and multiple models are supported. And one of the things that I want to highlight that now it supports continuous binary survival and multi-class outcomes. Nalin has worked on making this a comprehensive package, and he's going to talk about some of the other functionalities of integrated learner. Because of the time, I will actually wrap up here. I had a few more materials on horizontal integration, but for vertical integration, we can simply wrap up by saying that most of the published methods are early fusion. That includes fancier AI methods as well. Integrated learner is one of the late fusion framework that has some implementation and software available. Intermediate fusion is emerging now with several methods called cooperative learning, and some of the methods also developed by our lab called Bascoup and Jafar, which we are not covering, but happy to share details. If you are interested, do let me know if you have any questions on this intermediate fusion. But the take-home message is there is no clear consensus on the best fusion strategy. So we have seen that in our real data that early fusion sometimes win, intermediate fusion also wins, win in most cases, and late fusion is not something that you can just say that is out of the contention. So early fusion, late fusion, intermediate fusion all have their advantages and disadvantages. So I'll skip the part for horizontal integration and some of the diagonal integration stuff. Mostly I will start with here, and I will obviously share the slide deck. So there are several open problems in multimodal integration. There are some views that mention that there should be some ordering how you do the fusion. So there are some methods that try to respect that ordering, like DNA to RNA to protein type of thing. Like you should be doing integration in a hierarchical manner. And you can see that there are multiple ways you can do that. And it's a combination of sometimes imaging and omics, and imaging, you first need to extract features, so on and so forth. So it's a pretty open problem, even after so many years of development. And diagonal integration remains one of the areas where many open problems still exist. And you can see from the slide deck some of the examples in diagonal and horizontal integration. And one of the other open problem is, as the number of modalities are increasing, how do you select modalities? Like, can you actually drop some modalities at the very beginning based on some modality selection? So as you remember, possibly know from single view literature that we are interested in feature selection within views or within omics. Again, I'm using the term view as omics modality. These are all similar terms. Just wanted to make sure we are not using different terminology. So view is same as modality, same as single omics. So if you have multiple views and you want to have a situation where you don't want to model everything because you have collected the data, that comes to the problem of modality selection. It's also an open problem, and there is a way to do that based on this paper. It's called ablation analysis. It's very popular in computer science, where you systematically remove one modality at a time or include one modality at a time and see what is the performance drop when you do a various combinations. So that's one open problem that remains. And when it comes to bioconductor, and we are going to switch to Nalin's tutorial very soon, you are all familiar with summarized experiment. And obviously that was not completely useful for multiple assays. And that's why multi-assay experiment came about, which has this particular sample metadata that connects all these different assays together. And you can assess individual modalities as well as extract a set of subjects using specific functions within this container, or methods as you call it, I think, within multi-assay experiment. So we are going to see how to start with individual CSV files or maybe our data file, and then construct this multi-assay experiment, and then use integrated learner for a prediction problem. And before we move there, I just want to highlight one more thing is that, no, I think this is possibly it. And I think this is the last slide I wanted to show that think of a single omics. Any of your single omics is possibly high dimensional, depending on what data type we are talking about. It could be count, compositional, and there could be thousands of other challenges that already exist. And we already know that in the single omics setup. And that's why you have so many tools for zero inflation or how to model counts and library size and normalization. We all know all of that. So you can think of multimodal integration as times k and times s, depending on what you are trying to integrate. So if you have k number of modalities, you can simply think of this as k-fold more challenges or s-fold more challenges when it comes to multimodal integration. So I'll possibly end with that. And I think between now and Nalin's tutorial, we can also see if there is any question. I think I saw something in the chat. Okay. Okay, I will possibly stop sharing now, and I will answer the questions. And Nalin, you can set up the tutorial.
Yes, I'll just do that. Yes.
There are two questions. One is, how do we know that the relationship discovered between two data sets in diagonal integration reflect biology rather than similarities created by the integration method? So first thing is, in the diagonal integration, you cannot have any relationship meaningfully captured before you do the so-called alignment or harmonization. So that's step one. So if the question is, how do we know the relationship discovered? I'm assuming that the relationship discovered is after the alignment and after the diagonal integration, right? Is that fair? Mm-hmm. Yeah. So once you do the alignment, you have assumed that these two data sets are aligned and at least one of the variable or one of the axis. So depending on how good your alignment is, your biological conclusion is going to differ. So there are several methods that uses the concept of latent variable or some sort of, I think latent variable is the best term here, that you assume that there is a latent variable like something like a PC. Let's say I have two data sets, they are not aligned, but I want to project it on the principal component, maybe PC1. In some cases, the assumption is that they agree on the principal components at the latent space, because that's the assumption you have to make the things work. In those situations, it will be very difficult. So there is no such way that you can possibly know, because it's based on latent factor. But in other cases where you clearly know that you are doing the alignment at a level that is more interpretable. So one example is, typically, if you have subject and sample where the samples are not matching, for example, you have cells within subject, maybe spatial spots within another modalities, again, the same set of subjects, you try to do it at the subject level. So when you do that, you somehow first represent the data at the subject level. For the single cell, maybe you can aggregate the cells. For spatial spots, you can do some sort of spatial smoothing and then come up with the subject level representation. Then you do the diagonal integration. So those are much more reliable. So to answer your question is, if you do the harmonization in an interpretable manner, you are more likely to have biological interpretation, like subject level aggregation, for example. But if you do something like latent variable, I agree. If the question is, how do you know? You don't know. That in that case, you have to make a lot of assumptions. What makes diagonal integration different from simply analyzing two data sets separately and comparing the results afterward? the concept of integration is that you have um, somehow done some sort of joint modeling or joint analysis. Now this is more philosophical question. Like even this is you don't even have to do diagonal integration, just any integration. If I have let's say I have done differential expression analysis on uh, transcriptomics data, and then um, maybe differential abundance analysis on microbiome data, maybe on the same set of subjects or partially overlapping samples. Um, can we just take the results and try to compare them? That can be considered an integration or integrative modeling. But here, at least all the concepts that I have covered, here it means that you have jointly modeled the individual data in a single model or single framework. So that doesn't really count as integration if you are separately analyzing the data and then trying to get sense of the results based on the individual modalities. So that would be um, considered multi-omics integration, but maybe pseudo multi-omics integration if we have to invent another term. All right, let's do some practical hands-on in bioconductor. I think it's all yours, Nalin.
Okay, thank you for the lecture. I think you explained the multi-S experiment, summarized experiment. So I think I can start with that. So one of the things that is important for bioconductor packages is that it is dependent on summarized experiment if it's in a case of a single omic study. But in the case of a multi-omic study, it requires setting up a multi-S experiment. So to set that up, I'll just do a live coding session to start with. So first thing is that I would source the install file just in case to install all the packages. I think this should be really quick, but essentially our goal through this session would be to ensure that we have a data ready that can run on integrated learner, and the problem at hand is basically to see whether integrated learner can predict IBD samples versus healthy samples. And to do that, we utilize the IHMP dataset. The IHMP dataset, it contains two types of omics. One is microbiome and the other is metabolomics. So I'll start by reading the two omics now. So the first step would be to load the libraries. So I will load the three libraries needed. So the first library is dplyr. We need it because there are going to be cases where we join, we filter, and additional data frame operations. The other would be tidyverse. Again, for data manipulation, we need it. And then the third is the multi-S experiment library because essentially we want to build a multi-S experiment, so that is there in that particular library. Um, so yeah, I'll just run these. So all of these libraries are now installed. Now I think I can go to reading the datas. So the datas I currently have in the microbiome data is currently in a tree summarized experiment form, whereas the metabolomics data is in a matrix form. So I'll first read the microbiome data.
Nalin, is it possible to make it a little bit bigger
Okay,
so that
let me
everyone can see? Okay.
is it good?
Yeah, I think so. I mean, people can comment.
Yeah, so I'm basically loading this now. And then similarly, I can also load the metabolomic data set. So this is the microbiome data set, microbiome layer. This is the metabolomics layer. I'll just read them. Yes, so this should be done soon. after these are read, so now the thing is, all of these experiments, they tend to have their metadata stored in what we call a call data. So, call data essentially includes all of the information regarding the subject. So, for example, if you look at this, it contains all of the information regarding all of the samples included in the study. So, in this case, now the thing is that we first extract the metadata for each of the assays, because down the line, there will be a step where we need to ensure that the samples are consistent across both the omics layers. So, to do that, the first step is to get the call data for each of the assays. That is done using this function. Um, Similarly for the metabolomics, that would be done. Okay, so I'll just have a look at this quickly. Okay, yes. So now, another thing is that since we want to ensure that when we combine the uh, of two tree summarized experiments together, there is one single metadata for them. So to do that, we have to ensure that for both of these, the original metadata becomes null, so that there is no inconsistencies down the line. When we merge it, so let me do this real quick. Yes, so this will ensure that the metadata for both of the tree summarized experiments is null. And now the next step. So next step is essentially using all of these joining variables. These joining variables will ensure that we end up having the same samples across the two essays. So to do this, I will first ensure that I run this list once. Okay, yes. When this is done, now I need to ensure that the metadata for all of the two essays is consistent. And to do that, I will use an inner join, because we want only those samples which are present in both the omic layers. We do not want samples which are just present in, say, either the microbiome layer or the metabolomics layer and are not present in the other layer. So to do that, what we do is we use a merge by using all of the joining variables that I just declared above. And then we do all false because we only want the common samples. And to do that, we need to ensure that we do not make all true, because that would essentially join all the samples across the two layers. Then finally, so in all of these columns, there is a subject identifier called row.names, and we want to rename it to column name, uh, which will be a capitalized R row.names, to ensure, like, just for to create consistency across them. So that's done, and we end up having a metadata data frame, which just contains all of the common samples. So after having these common samples, we can first just do a check to see the number of samples here. And yeah, we end up having 385 samples and 38 columns. Okay. So now we need to ensure that both of the essays that we have, they have the same number of samples to do that. So in both the tree summarized experiments that we have, so the columns are your samples, whereas the row names, they tend to be the the feature name. So in this microbiome tree summarized experiment, I will end up having only those uh samples which are present in the metadata. Let's just run it, and then again, similarly for the metabo one, so after doing this, now I would like to check whether the number of samples across the uh two two tree summarized experiments or summarized experiments are consistent with the metadata to see this should be true. Yes, it is true. Um Similarly for the other one, we do that. Yes, so both of these are true now, I think. And so now we have essentially created two summarized experiments which are which have common samples, which was an essential step for integrated learners. So now we can go ahead and create a multi-S experiment. To do that, I will create a new object called MAE. Now, MAE multi as experiment here, we first need to declare experiments, which would be an experiment list, which would essentially include the microbiome layer, which would be our TSE, and the other would be a metabolome layer, which would include our metabo.c. Yes, after doing this, the next step would be to declare the call data, which would be the metadata we have. So let's see, let's have a look at this. Um, let's quickly have a look at this. Yes, so we end up having 385 samples across the two layers, and one is the microbiome layer, and the other is the metabolome layer. So in essence, we started with two individual layers. With unequal number of samples, and so the first step that we went through was essentially uh, creating the metadata, combined metadata, by using a couple of joining variables. We did an inner join there and we ended up getting a subset of the samples, and then using the original TS experiments and the metabolomic experiment, we got a uh, subset of the two datas, and then we combined it to form a multi-S experiment with having common number of samples. Any questions here? Okay, I think then I can go ahead with the integrated learner part now, I think. There is one in the chat. Do you have to use I'll be coming to that when I go into the deeper detail of running the integrated learner. There is a step that will be there where we transform the two different omics layer using transformations. So after having the MAE ready, there are a couple of steps that we will do. Since multi-omics data, they tend to have a lot of features, so we would like to reduce them, and we do that using two strategies. One is using a prevalence-based threshold, and the other is using a standard deviation based method where we check for each feature's standard deviation and see if it's less than a threshold, we drop that particular feature. So um coming to the first case, we essentially would be trying to calculate the prevalence of each of the features and then filter it. Prevalence is the calculation of basically how often a particular feature is non-zero in a sample. So we use that. Now to do that, since we have already made our MAE experiment, I will do this MAE microbiome. Then the Mia verse packages, they tend to uh, have a couple of good functions here. So subset by prevalent is what we will be using here. So we first input the layer that we want to filter, which would be the microbiome layer for the MAE. Then our assay type is relative abundance here because microbiome data is relative abundance. Yeah, relative abundance. And then we want to put a prevalence threshold. Prevalence threshold would be 0.1 in our case. And then we also want to give a pseudo count value. Sorry, not pseudo count value, but include dot lowest, make it true. So this would end up filtering the microbiome layer. Let me just have a look why it's just.
It looks like it cannot find subset by prevalent. Do you have?
Yeah, I
Oh,
think it worked right now. Yeah. Then again, metabolome. Here we would look at the NMR value and then we run it again. Okay, so here we have filtered out the number based on the prevalence of. So let's just have a look at the features. So this is 548 features for the metabolome layer, and this is and we end up having 128 features for the microbiome layer. So okay, so the first step was to do it based on to filter out features based on prevalence. We've done that. The next step would be to calculate the standard deviation to see how much the features vary across samples, and then features which have non-zero variance, we keep those, and once with variance near zero, we drop them. So to do that, we first calculate the raw data and then we create an ST column which contains the standard deviation values for each, and then we use matrix stats, row STS, yes, and put in the layer as the input. That would be assay of a MAE microbiome. And again, it would be relative abundance here. Abundance here and yeah, this would be the calculation. After that, We again do the same for the microbiome layer, metabolome layer. And this would be the NMR. Okay, so we've calculated the standard deviation for each of the layers, and now we just drop those which have a standard deviation less than 0.0001. So to do that, we do microbiome. I see a couple of questions. Yeah. Um, we do MAE microbiome, then raw data, MAE microbiome, $ST greater than 0.001. Yes. Similarly for the metabolome, this would come here. Yes, so now we have done the filtering stage. We've reduced the dimensions of both of our omics layers, and now what we need to do is we need to normalize the data. So to do that, so microbiome data tends to be highly compositional. That is basically, since it uses relative abundance. So in a case where the relative abundance of one of the organisms in the data, it increases, the other organisms' abundance will decrease, because all of the abundances of organisms in the sample, they tend to sum up to a fixed number. So to counter that, we for microbiome layers, we would do a CLR transformation, which is a central log ratio transformation. Whereas for metabolomics tend to be highly right skewed. In that case, we would do a log 10 transformation on them. So to do that, for the microbiome layer, we again use a function from the Mia verse package, which would essentially do this. MAE microbiome. We give the assay type. There are a few. Let me just have a look at the questions. Yeah, I will just add the scripts to the GitHub, and or I could just paste it there. That would be there. Relative abundance. Abundance. And then we would put the method as CLR in this case. And then we would have to add a pseudo count here, because there can be cases where the calculation ends up being close to zero. For those cases, we need to ensure that there is a pseudo count. So for the microbiome layer, it is done. Now coming to the metabolome layer. Here, since we want to do a log 10 transformation, our method will change up to be log 10. And then pseudo count will be 1, because log of 0 does not exist. and this would be NMR. Yes. Yes, so now we have done all of the pre-processing needed. We've filtered out to only have a highly variable features or a highly prevalent features. And now we have normalized both of our layers. So the next step is that so in machine learning, it is a general practice to have a training and a testing data set. So now we would go ahead and try to create those. So to do that, I would I would store the metadata of our MAE into a different variable so that we can work on this and do not modify the original metadata. Let's just have a look at the metadata there. Um, Yeah, okay. So to run integrated learner, we need to have a Y column, which would be our outcome column. And to build the outcome column, we would use the study function here. Study column has these entries, which would be IBD patients and healthy patients. A control patient. So we would create a Y column out of the original metadata using this call, which is hash dot numeric cd dollar study condition equal IBD. Um, we did this, and now we can have a look at the table to see how this looks like. Yeah, so controls have ended up becoming 0 and IVD samples are 1 now. So one of the things with integrated learner is that you cannot give you cannot have a non-numerical labels, especially for binomial cases. So it is an essential step to convert it into a numeric 0, 1 case. Now the next step is that IL also needs a subject identifier. So we will go ahead and try to create it right now, which would essentially be just this. Um, just creating a new variable using the original existing subject ID variable. We have done that. Now the next part is to split up the data set into training and a testing data. So to do that, again, CD, I have it already stored. So let's do this. Subjects, we need to have the subject ID stored because we would essentially be using that to break it down into a validation or a training data set. So that would be all of the unique IDs of the subject ID. Now, again, we try to create a list of validation subjects, what would be using again Meaverse based function, which is called sample, which essentially takes the list of our subjects and the number, which would be we are doing a 80-20 split. 80% of the samples go into the training data set, whereas 20% of the samples go into a testing data set. So that would be 0.2 into the length of the subjects.
Yeah, I want to quickly mention that in many cases, you may have the validation set already. So in that case, you don't have to create this validation set, and you can simply use it in the validation data as your validation set, and it will run as it is.
Yes. I forgot to set a seed, so I should have done that. I'll do that. And then, yeah, I got a list of validation subjects. Now, the next step is to ensure that I have separate data sets for the training and the validation data sets. So I would quickly create another list of just having samples which are in the validation subjects, which would essentially go like this. ID percentage and percentage valid subjects. Yes, this would be okay. This is essentially true false. So now I would do this. Train samples would be row names, MAE, row name, sorry, CD, percentage in valid. Yeah, so here I am using the row names of the metadata and filtering out all of those samples which are in the validation data set. So I do this. Similarly, I will do this again, but this time since I have a list that includes all of the samples which are going into the validation list, I build, I run this and then yes, so I have now built up two list of training samples and validation samples. Now all I need to do is create two MAE objects, one for the training and the other for the validation. The MAE train will be created as MAE train samples. Here I'm essentially storing all of those samples which are in the training data set, and similarly MAE valid, which would be all of the validation samples. Yes, so this is done. So we have created a validation data set, we've created a training data set. Now, we on these data sets, utilizing these, we can quickly call integrated learner, which is here. So we need to give the training data set as well as the validation data set here. Then this would be our validation data set. After that, we also need to declare the experiment to be utilized. In our case, it is the microbiome and the metabolome. So uh, there are multiple essays. If you look at each of these experiment types, so we need to also mention which to utilize. We would utilize the CLR based for microbiome and the log 10 based for the metabolome. Now we also need to declare our outcome column, which would be Y in our case. Um, and then we also need to give a subject ID call, which would be our subject ID function. And then we can also need to mention the number of folds for cross validation. Cross validation, as Dr. Malik mentioned, is a crucial aspect of late fusion. And for now, I mean, I'm just going for two because we wanted to run it quickly instead of doing five. Five might take a bit more time. And then we have a base learner, which is in this case, I am running random forest. It can be any other uh, model as well that superlearner supports. Then we also need to declare the meta learner strategy, which would be SL NNLS AUC. Um, then after that, we also want to run both the stacked version, which is the late fusion version, and the early fusion method, which is the run concat function. Both of them, we need to set them to true and then run. Let's see. Let me just quickly grab this from one of the codes instead of writing it all again. Yeah. Mhm. CD. What happened?
Can you make sure the name is subject ID that in the MAE?
Yeah, let me just have a look once. Mm-hmm.
So it's a different name, right?
Yes.
So you have to put subject underscore ID.
Yes.
So those who are following, this is required because that's how integrated learner separates the modalities and do the individual level modeling. So if you don't give the subject ID, you don't be able to do that.
Hmm.
Yeah, I think somewhere this subject ID has been lost. Maybe
Let me just quickly grab all of this from this part and then run it. Ha. Okay. I'll
oh, this
just
ME train was not built.
No, it was, but somehow it just went away. I mean, I'll just run this script that I was going to put up in the uh GitHub. It essentially does all of the same steps, and yeah, I think I might have missed some step here. I'm not sure.
Yeah, but it's now running. It's good.
Yes. Yes.
Yeah. So unfortunately for multi-omics data using Bioconductor, you have to go through these pre-processing steps before you can even run the pipeline, which is a little bit of pain. We are trying to make it simpler as we go along in the software updates in future. Okay, is it running a really high number of cross-validation? Why it's taking too long?
It is running 5. Let me just pause it and put it to 2.
Okay, yeah. So it should be really quick now. Let's see. Why are you not seeing any?
Yeah.
Okay, now you're seeing. Okay, great.
Yeah, so the output includes the time it took to fit the model. It is inclusive of all of the individual layers model and then the concatenated layer. So coming to the result here, we see the AUC output for the training data. Each layer has its own AUC output, which is 0.7 for metabolome, and microbiome has an AUC output of 0.63. The STAT model, which is the late fusion model, it has an AUC of 0.71, whereas the concatenated model, the early fusion model, it has an AUC of 0.7. Now coming to the testing data, the testing data is based on the output is based on the holdout set, which includes the uh, for the metabolome, it has an AUC of 0.89. For microbiome, it has an AUC of 0.74. For the STAT model, the AUC increases to 0.92. And for the concatenated model, it is an AUC of 0.9. And we also get the weights by for the late fusion based result. For metabolome, the weight is 0.713, whereas for microbiome it is 0.287. There are different aspects of the integrated learner result that can be accessed. So model fits here is essentially all of the layers. Model, you can access it to get the importances. I will also show a code which calculates the feature importance for all of the late models, or for late fusion. And then there are other outputs here as well. Y-hat test is the prediction based results. And there are other attributes to the integrated learner call. So, for example, let's say you want to drop a layer from a multi-assay experiment which is not performing up to the mark. For example, here, let's say we had another layer of functions and it performed poorly. It had an AUC of less than 0.5. We can put a threshold there of 0.5 and drop that particular layer. And there are also other metrics that one can get from here. For example, let me just print this out. So yeah, so here we also calculate the accuracy and balanced accuracy along with the AUC for individual layer as well as late fusion and early fusion based methods. So now coming to the feature importance aspect. Another function of integrated learner is that you can also get AUC ROC curves by calling this function integrated learner plot learner fit. And this yeah, for now, because we have fold as 2, it might not fit, but for 5 it does. So if you can so after uploading it on GitHub with fold as 5, you will be able to run it and generate the ROC curves. And then the other aspect of integrate learner is that it provides each layer's feature importance score, which can be multiplied by weights to get a score of each of the feature for late fusion, and then you can visualize these features. Let me quickly open this up. Wait. This might not be, I'll have to change it again.
Okay, I think it's okay, Nalin, given that we are 15 minutes above time already. So maybe you can simply wrap up. And so good thing about the integrated learner tutorial is that you can follow along once you know these basic steps, which we wanted to show. And it can support continuous survival and multi-class outcomes if you have. And there are various ways to tune the model, for example, cross-validation fold, in validation set versus not, and different base learner, different meta learner, and several other considerations. So obviously not everything is possible in this particular lecture, but feel free to reach out if you have any questions to us. I'm going to provide my email address just in case you have any follow-up question on any of the part of the lecture. And I will also share the slide deck with the core team after this. And Nalin, you can also put your email because you might be getting questions from the software side of things.
Sure. I'll do that as well.
Yeah. Thanks again, everyone. I think just want to thank the core team and everyone for joining today. And thanks for the great questions.
Yes, thank you so much, Dr. Mallick and Nalin, for creating time and giving this lecture to Bioconductor Africa Seminar Series. We are very glad that you could make it. And yeah, so thanks everyone for joining. I will archive the notes. Hopefully Nalin will paste the code there, or you'll just clone from GitHub and you'll be able to practice at your free time. I have also pasted both of their emails on the Frama pad if you want to check them out later. or contact them with further questions. At this juncture, I would like to say next month we are going to be moving to single-cell RNA-seq analysis with Jack Cerise. So we'll be happy to have you again, and maybe we might, if there's sufficient interest from the community, we will be able to re-invite you, Dr. Mallick and Nalin, just to, maybe to wrap up that practical part so that, people have those practical skills. Thanks again for joining. In case you've not joined our mailing list, that's line 94 of the Pad. Also connect with us on Zulip, that's line 92 of the Pad. And yeah, this session is made possible by funding from the Chan Zuckerberg EOS funding cycle 6. And yeah, thanks so much for joining, everyone. See you next month.
Thank you.
Thanks.
Thank you.